Hello everyone, if you are preparing for NEET, then this video is for you. In today's class, we will solve highly important MCQs from biotech and research application, focusing on exam trending concept and previous year pattern. This session will help you score better with smart preparation. Let's get started. Okay, so the first question, it says the critical research area of biotech include all of the following except. So the critical research area, it says the critical research area of biotechnology field, it includes what? It includes best catalyst in the form of improved organism, usually a microbe or pure enzyme, creating optimal condition through engineering for a catalyst to act, downstreaming process technology to purify protein or organic compound. But in question we should circle out what is given. Except, they say except. So except means we cannot develop newly genetically modified microbes that can be used as bioweapons. We don't want bioweapons. Okay, so biotechnology is not used for development of bioweapons. Okay, next question. Increased yield of crop during the green revolution was due to what? Increased yield of crop during green revolution, it was due to use of improved crop variety, use of better management practices, use of agrochemical, that is fertilizer and pesticides. Okay, all the three options are correct. Next, it says any part of the plant taken out and grown in a test tube under sterile condition in a special nutrient media for generating new plant is called what? Explant. The term totipotency refers to a capacity of what? It refers to a capacity of the cell to generate the new whole plant. The most important advantage of micropropagation is what? It is rapid production of large number of genetically identical superior plant. Micropropagation is propagation of the plant in vitro. Remember, propagation of the plant in vitro. Propagation of the plant in vitro. The plantlets produced during plant tissue culture are called soma clone. Why are they called soma clone? Soma clone means what? They are genetically identical to the original plant from which they were grown. Soma clone variation are seen in the plant that have been produced by plant tissue culture. Soma clone variation are seen in plants that have been produced by plant tissue culture. Next, a crop gets infected with a certain virus and the farmer of the area suffers immense financial loss. You have been asked to help the farmer and you decide to improve the crop by raising virus-free plant through plant tissue culture. Okay, the explant you will choose will be meristem. Meristematic tissue, we already have read this in what? In chapter number 6 of class 11. Okay, next, the first step to achieve somatic hybridization in the plant will be what? The first step to achieve somatic hybridization in the plant will be converting plant cell into protoplast by cellulase. The first step to achieve what? To achieve the somatic hybridization. To achieve somatic hybrid, we want somatic hybridization. So what will be the first step? The first step will be converting the plant cell into protoplast by cellulose. Now what is protoplast? Here the next question comes. What is protoplast? Protoplast is a plant cell without a cell wall. Protoplast is what? It is a plant cell without a cell wall. Okay. Next, given below are few statements regarding somatic hybridization. Choose the correct statement. Let's choose the correct statement. First statement, it says that protoplast of different plant cell of the same plant are fused. Protoplast from different cell of different species can be fused. Treatment of the cell with cellulose and pectinase is mandatory. The hybrid of the protoplast contains character only of one parental protoplast. No. Correct statement. First of all, we will mark this. They are saying correct statement. Here where the NEET aspirants make mistakes. These are silly mistakes. Okay, now the protoplast of the different cell of the same plant are fused. Protoplast of different cell of same plant are fused. No, this is a wrong statement. Different cell of same plant means what? Protoplast can be fused. Okay, a hybrid cannot be formed. Next is protoplast from cells of different species. Yes, this is a correct statement. This is a correct statement. Protoplast from cells of different species can be fused. Yes, this can be fused. Okay. Next is what? We will treat the cell, treatment of the cell with what? Cellulase and pectinase. Okay, cellulase and pectinase, it is mandatory. Yes, this is also a correct statement. And the hybrid protoplast contain character. Hybrid means what? From both the parents, not only one parent. So this is a wrong statement. Okay, next question. What does it say? All of the following are possible today with the use of the GM crop, except what? Again we will mark except, the word except. So what is possible today with GM crop, right? So we will say more tolerance to abiotic stress. Yes, this is possible. Alternative resources to industries. True. Then in the increased efficiency of mineral usage by plants. True. Fixation of the atmospheric nitrogen. No, we cannot fix the atmospheric nitrogen by what? Genetically modified crops. We cannot fix the atmospheric nitrogen. So this is a wrong statement, and this is what they have asked in question. Next question. The GM crops have been modified with various traits. Golden rice have been developed by International Rice Research Institute for what? GM crop have been modified with various traits. Okay, GM crops what? They have been modified with various traits, and golden rice has been developed by the International Rice Research Institute. That is, it can be said as IRRI. RIRRI. Okay. So GM crops, they have been modified with various traits. Now golden rice have been developed by the International Rice Research Institute. Why? For what? For what purpose? For the improved nutritional value. Okay, next question. Consider the two statements. Use of the genetically modified crop can be possible solution for minimizing the use of the chemical and the fertilizer for detrimental for the environment. Yes, this is a correct statement. We can use the GM crop for minimizing what? For minimizing the use of pesticides, the chemical, different harmful chemical, isn't it? GM crops do not require any fertilizer. No, they require fertilizer. Okay, they also require fertilizer, but we can use organic fertilizer. Okay, so statement 1 is correct. This is a correct statement. This is a true statement, and this is a false statement. Okay, next, BT cotton is not what? Again we will mark the word not. Okay, BT cotton is not what? The BT cotton is not a GM plant. No, BT cotton is a GM plant. BT cotton is insect resistant. BT cotton, a bacterial gene expressing system. Yes. Resistant to all pesticide? No, they are not resistant to all pesticide. Next, Bacillus thuringiensis, that is the BT strain, have been used to design novel what? BT is only for insect. Remember, they are only for insect. Okay, they are related to insects. Okay, so what we will say? Bioinsecticidal plants. Remember it like this. Okay, next, some strain of Bacillus thuringiensis produce protein that kill certain insects, such as what? Such as Lepidopterans. They are also insect, using tobacco budworm and armyworm. Coleopterans, including beetle. Dipterans, including flies and mosquitoes. So this some strain of Bacillus thuringiensis, they produce protein that kills certain insects. So it may come they may alternate the question, they may modify the question, and they may say that what are the lepidopteran insects name? The lepidopteran insects name are tobacco budworm and armyworm. Next is Coleopterans is beetle. Coleopteran will be beetle, and Dipterans. Dipterans will be flies and mosquito. Flies and mosquito. Flies and mosquito. So leptidodurans. 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 It will be what? Tobacco related. Okay. Leptidodurans will be tobacco what? Tobacco budworm. Tobacco budworm. Please manage with my handwriting. Tobacco budworm and what? And armyworm. Tobacco budworm and armyworm. Tobacco budworm and armyworm. And coleopterans. From coleopterans we will see. 1 second guys.
Hello. Nah.
Sorry guys for the disturbance created. Okay, so leptidodurans will be tobacco budworm and armyworm, and next is coleopterans. Next is what? Coleopterans. Coleopterans example what? I said beetle. Coleopteran will be beetle. Coleopteran, beetle. Coleopteran, beetle. And dipterans. Dipterans will be flies and mosquitoes. Flies and mosquitoes. Flies and mosquitoes. So statement 1, 2, 3 all are correct. Next is Bacillus thuringiensis. That is the BT. Full form of BT is Bacillus thuringiensis. Bacillus thuringiensis, it is significant for biotechnology because why? It produces BT toxin. It produces what? BT toxin, which is effective against insect pests when introduced into the crop. It produces what? It produces BT toxin. There is a toxin named BT toxin which is effective against, as I said, see BT means what? Insect. Okay, so effective against what? Insect pest and introduced into the crop. When introduced into the crop. Done. Next, consider the given two statement. Spores and crystalline insecticidal protein produced by BT thuringiensis are now used as insecticides in organic farming. Spores and crystalline insecticidal protein produced by B. thuringiensis are used as insecticides and organic farming. Yes, this is a correct statement. Now let's see the second statement. Because of their specificity, these pesticides are regarded as environmental friendly with little or no effects on human, wildlife, pollinator and most other beneficial insect. So this is an assertion reason question. First of all, both the statements are true. Now we will see if statement 1 what? Justifies statement 2. Yes, statement 2 justifies statement 1. We have to check this. So yes, because what? BT toxin, it is only harmful to what? It is only harmful to the insect pest of the crops, not to what? It is environmental friendly. It is not harmful to human, wildlife, pollinator and other beneficial insect. Only specific insect. Okay, only specific insect. I have mentioned the name of the insects before. That is leptidodurans, tobacco budworm and armyworm. That is coleopterans from beetle, and dipterans, that is flies and mosquito. Okay, this much only. These insects are mentioned. So we can say that both the statement 1 and 2 are correct, and statement 2 explains statement 1. Next is the trigger for activation of the toxin of Bacillus thuringiensis, that is the BT. The trigger for activation, when the toxin is released. Okay, they are asking this question, when the toxin become activate, because this is generally inactivate. Okay, this is generally inactivate, but when they reach the alkaline pH of the gut, they become activate. Okay, alkaline pH of the gut of what? Gut of the insect. Alkaline pH of the gut of the insect. Next is identify the incorrect statement. Again we will mark the word incorrect. Okay, now let's see. BT thuringiensis, that is B thuringiensis, forms protein crystal during particular phase of its growth. Yes, this is true. The protein crystal contain a toxic insecticidal protein. Yes, this is also true. When an insect ingests the inactive toxin, it is converted into an active form of the toxin due to the acidic pH of the gut. Now only we have studied that it is the alkaline pH. It is the what? It is the alkaline pH, not the acidic pH. So this is a wrong statement. They have asked about the incorrect statement. So this is a wrong statement. Next it says the activated toxin binds to the surface of the midgut epithelial cell and create pores that cause swelling and lysis and eventually causes the death of the insect. Yes, this is also a correct statement. Only the incorrect statement is that it will be what? It will be not acidic, this will be alkaline. That is alkaline or basic. Okay, done. Next question. Corn borer. Corn borer can be controlled by the protein. Let me check. It is written here. Encoded. This will be encoded. Let me complete it. Encoded. Okay. So okay. Corn borer can be controlled. They can be controlled by the protein encoded by what? Encoded by Bt gene. That is cry1AB. When corn borer, then it will be cry1AB. Okay. So remember it like this. Okay. First let me complete question number 24. Then I will give you a short trick. Protein encoded by cry1AC and cry2AB gene in Bt help in control of what? Cotton bollworms. Cotton bollworms. Now let me give you a trick how to remember this. How I remember it, remember this is say there will be three kind of gene. Okay, there will be three kind of gene. Three genes. What are the three genes? 1, 2, 3. One is cry1AB, cry1AB, cry2AB, cry2AB and cry1AC. This much is clear. Three genes. Okay. Now see cry1AB, then cry2AB and then what? Cry1AC. So the first one goes for cotton borer. Cotton borer. B-O-R-E-R. Okay. First one goes for cotton borer and the last two genes cry2AB and cry1AC. These two goes for cotton bollworm. Cotton bollworm. Please manage with my handwriting. I am really very sorry for my handwriting. Okay. So protein encoded by cry1AC and cry2AB gene of Bacillus thuringiensis help in the control of cotton. So from now on, remember this trick that gene are of three types. Cry1AB, see first come first serve. We already know. So cry1AB, then cry2AB and then cry1AC. Then comes cry1AC. Okay. So cry1AB, it will be for cotton borer. First is cotton borer, and cotton bollworm. Bollworm is a large name, isn't it? Bollworm is a large name, so it goes afterwards. And when it goes afterwards, it will go with two genes, that is cry2AB and cry1AC. Cry2AB and cry1AC. Done? Okay, next. Consider the given two statement. Statement 1 is Bt cotton contains gene from the bacteria Bacillus thuringiensis which produce cry protein toxic to specific insect pest. And statement 2 says the cry protein formed in Bt cotton dissolve the gut lining of the insect leading to their death when they feed the cotton leaves. Statement 1 is I don't know, I did not get statement 1, but statement 2 when I read it I got it immediately. So statement 2 is true. Let's read statement 1 again. Bt cotton contains gene from the bacteria Bt. Bt cotton contains gene from the bacteria Bt, okay, which produce cry protein. Which produce cry protein? Yes, we have already seen three type of protein by gene, that is the Bt gene, okay, Bt gene. So Bacillus thuringiensis which produce cry protein toxic to specific insect. Yes, this is very toxic to only a specific insect pest. This is also a true statement. Both the statements are true. Both the statements are true. Sorry. Both the statements are true. Next, a transgenic tobacco plant was created to make it resistant to what? Remember, a transgenic tobacco plant, it was created why? It was created to make resistant to a nematode, that is Meloidigni incognitia. Meloidigni incognitia. Okay. So nematode will be what? Meloidigni incognitia. Next is what? RNAi. RNAi as a mechanism of cellular defense takes place in what? It takes place in all eukaryotic organism. It takes place in all eukaryotic organism. It takes place in what? All the eukaryotic organism. RNAi is what? RNA interference as a mechanism of cellular defense takes place in what? It takes place in all the eukaryotic organism. Nematode Meloidigni incognitia. It is specific gene were transferred to tobacco plant using what? Using agrobacterium meditation. Agrobacterium. This specific nematode, what did it do? These were transferred to tobacco plant. These were transferred to the tobacco plant using what? Agrobacterium mediation. Agrobacterium mediation. Okay. It was created to make a transgenic tobacco plant was created. It was a transgenic tobacco plant. It is a transgenic tobacco plant. It was created to make it resistant to a nematode, Meloidigni incognitia. Okay. Now this nematode specific gene were transferred to the tobacco plant. This was what? This nematode was created. Why was it created? It was created for created to make resistant to the transgenic tobacco plant. To make resistant transgenic tobacco plant. Nematode Meloidigni incognitia specific genes were transferred to tobacco plant using what? Agrobacterium medication. Okay. Next. RNA interference involves silencing of specific mRNA. RNA interference involves what? Silencing of a specific mRNA. How? By a double-stranded RNA that binds to it and prevents its translation. RNA interference involves silencing a specific mRNA. RNA interference means what? RNAi. Okay, RNA small I. So RNAi involves silencing of a specific mRNA. Silencing of what? Silencing of a specific mRNA. Why? By a double-stranded RNA. How? By a double-stranded RNA that binds to it. By a double-stranded RNA that binds to it and prevent. Why? To prevent its translation. Because this is what? This is harmful for the tobacco plant. So we need to silence the gene. So for silencing, what we will use? We will use a double-stranded RNA. RNA interference involves silencing a specific mRNA by a double-stranded RNA that binds to it and prevents its translation. That prevents it what? Translation, not transcription. Okay, it's translation. Next, identify the incorrect statement regarding the use of RNAi for creating transgenic tobacco plant resistant to Meloidigni incognitia. Now, they have said incorrect statement. We have to find out for creating regarding the use of RNAi for creating transgenic tobacco plant resistant to Meloidigni incognitia and nematode. Okay, first it says Agrobacterium tumefaciens was used to transform the plant cell. Yes, this is a true statement. We have already read here. See. Agrobacterium medication. Nematode, how was it transferred? By the injection of agrobacterium medication. Okay. Now. Double-stranded RNA was introduced into the plant cell. Double-stranded RNA was introduced into the plant cell. Third option says double-stranded RNA was formed in the plant cell that initiated RNA interference. Fourth option says that a specific mRNA of the nematode was silenced with the help of double-stranded RNA. Okay, So now they say that double-stranded RNA was introduced into the plant cell. No, it was not specifically introduced from outside. Not from outside. It was already biological, okay? RNA interference was already a biological process which was already formed in the plant cell. It was beforely formed into the plant cell which initiated RNA interference inside its own body. Okay, clear? So this is a correct statement, this is the wrong statement. For here, incorrect statement will be option number 2, and this is already correct. It was silenced with the help of double-stranded RNA. Clear? Done. Okay, let's continue. The source of complementary RNA for RNAi in an eukaryotic cell could be what? It could be an infection by the viruses having RNA genome. It could be what? Infection. The source of complementary RNA. The source of complementary RNA for RNAi in a eukaryotic cell. The source of complementary RNA for RNA interference in a eukaryotic cell could be what? It could be an infection by the virus having RNA genome. Okay. Mobile genetic element that replicate via an RNA intermediate. Mobile genetic element. Yes, this is also true. Mobile genetic element that anything related to, remember, for RNA interference. For RNA interference, anything related to what? Anything related to the RNA effect, RNA gene effect. Okay, anything that affects RNA gene, it will be what? It will be it will lead to RNA interference. Okay. So the source of the complementary RNA for RNA interference in a eukaryotic cell could be what? It could be an infection of the viruses having RNA genome, not DNA. DNA will not have any kind of effect on this particular RNA interference. Okay. Next is mobile genetic element that replicate through an RNA intermediate, and integration of the phased DNA. Anything related to DNA, it will not have any effect, or it will not lead to what? RNA interference. Next, a biological process in which RNA molecule inhibit gene expression or translation by neutralizing targeted mRNA molecule is known as what? It is known as RNA interference. So what is RNA interference? RNA interference, it is a biological process which happens inside the body only when they see any kind of distransformation or any kind of breakage in the RNA molecule, any kind of defect in the RNA molecules. So what happened? The body itself inhibits that gene expression or the translation. Translation is what? Gene expression. By neutralizing what? By neutralizing the targeted mRNA. They will target the particular mRNA molecule and they will silence that particular mRNA. Okay, from translation. Now what is RNA interference? RNA interference involves silencing a specific mRNA due to the complementary double-stranded RNA molecule that binds to and prevent translation of the mRNA that is silencing. This is a true statement. It has been used to create transgenic tobacco plant resistant to nematode, Meloidogen and incognitia. Meloidogen incognitia. I don't know the pronunciation. Please pronounce it by yourself. So this will be what? RNA interference. Both the statement are what? Both the statement are true statement. This was RNA interference was first time seen in transgenic tobacco plant, and what? And the definition we already read it. So both the statements are true. Next is what? Next is biotechnological application in medicine. Biotechnological application in medicine. And let's see. The first drug produced using rDNA technology is used to treat what? Diabetes, of course. Next, the two polypeptide chain of insulin protein are linked together by disulfide bonds. Next, C-peptide of human insulin is removed during maturation of the proinsulin to insulin. Removed duration. C-peptide of the human insulin. C-peptide of the human insulin is removed. C-peptide of the human insulin is removed how? When and how? removed during maturation, during the maturation of pro-insulin to insulin. C peptide of the human insulin is a part of the mature insulin molecule responsible for it is removed. C insulin has to be removed. It has to be removed during maturation of the pro-insulin to insulin. Next is the main challenge for the production of the insulin using rDNA technology was what? The main challenge, what was the main challenge? Getting insulin assembled into a mature form. It could not form the, the main challenge for the, by rDNA technology, everything was fine until the maturation phase came. They could not mature the insulin, okay? They could not mature the insulin by rDNA technology. So the main challenge for the production of the insulin, by rDNA technology was what? It was getting insulin assembled into a mature form. Okay, so the next question. Okay, so how many of the given statement regarding insulin are correct? We will see how many of these given statement regarding insulin are correct. First is all diabetics require insulin from the very beginning. Insulin can be given orally to diabetics. Insulin extracted from the pancreas of the slaughtered cattle and the pigs caused some patient to develop allergy. Recombinant insulin was first prepared by Indian pharmaceutical giant Ranbaxy. The two polypeptide chain in the final structure of the insulin are joined by disulfide bond. DNA sequence corresponding to A and B chain of the human insulin were introduced in the plasmid of the E. coli to produce insulin chain for preparing recombinant insulin. Okay. We have to find out what? We have to find out how many of these given statements are correct. First statement says all diabetics require insulin from the very beginning. From the very beginning? No. All diabetics do not require insulin from the very beginning, isn't it? So this is a wrong statement. First one. Second one. Insulin can be given orally to diabetics. No. Insulin cannot be given orally to diabetics. Why? Because insulin is what? Insulin is a, let me write it here. Insulin is a protein. Insulin is a protein. When we will ingest it orally, okay, when orally given, what will happen? It will be digested. It will be digested. Okay, and we don't want this. Digestion, we don't want this. Okay, we don't want this. So, insulin can be given orally to diabetics? No, it cannot be given orally to diabetics. It can be only given by what? Injection. Next, insulin extracted from the pancreas of the slaughtered cattle and the pigs caused some patient to develop allergy. Yes, this is a true statement. This is a true statement. Fourth, recombinant insulin was first prepared by Eli Lilly, not Indian pharmaceutical giant Ranbaxy. No. Next, the two polypeptide chain in the final structure is joined by disulfide bond. We already know insulin is the two polypeptide chain is joined by what? Disulfide bond. DNA sequences corresponding to A and B chain of the human insulin were introduced into the plasmids of E. coli to produce what? Insulin chain for preparing recombinant insulin. Yes, this is a true statement. DNA sequence corresponding to A and B chain of the human insulin were introduced in the plasmid of the E. coli to produce insulin chain for preparing recombinant insulin. So, 3 statements are correct. Next, to produce human recombinant insulin, Eli Lilly prepared 2 DNA sequences corresponding to what? Corresponding to A and B chain of the human insulin and introduced them in the plasmid of the E. coli to produce insulin chain. Prepared 2 DNA sequences corresponding to A and B chain of the human insulin and introduce them in the plasmid of the E. coli. Plasmid of the E. coli to produce what? Insulin chain. To produce insulin chain. To produce insulin chain. Next, insulin earlier extracted from the pancreas of the slaughtered cattle and pig was not effective in bringing blood glucose level down towards normal range in diabetics, caused some patient to develop allergy and other type of. It was effective. It was not that it was not effective. This is a wrong statement, but this is a true statement. It caused some patient to develop what? To develop allergy and other type of what? Other type of reaction for the to the foreign protein. So option 2 is only correct. Next, the first recombinant therapeutic product approved for human use was human insulin. It was first recombinant. So remember, first recombinant therapeutic product which was approved for human use was human insulin. Next, the plasmid of which of the following bacteria was used to produce human insulin? Escherichia coli, E. coli was used. Next, and can you say me in the comment section what for Agrobacterium tumefaciens, what was used? What was used? Can you tell me in the comment section? Okay, next. What is an advantage of the recombinant insulin over insulin obtained from the animal sources? Okay, I will say the answer of question number 42, option number 4. I'll say it in the end of the video. Now let's move to question number 43. What is the advantage of the recombinant insulin over insulin obtained from animal sources? Advantage of what? Advantage of the recombinant insulin. We have to find the advantage of the recombinant insulin over the insulin obtained from animal resource. Obtained from the animal resource. First option. Recombinant insulin is cheaper to produce and more ethical. What is the advantage? We have to find the advantage, isn't it? It is identical to the human insulin, reducing allergic reaction and increasing efficiency. It can be orally ingested instead of injected. It has a longer lifestyle. No. Only option 2 is correct. Only option 2 is correct. That's why we are not using what? Reducing allergy. It reduces allergy, so it is more preferable nowadays. Instead of using the insulin from what? From pancreas of the slaughtered cattle and pig. Okay. Next. Okay, so let's continue with question number 44. It says, human insulin produced through recombinant DNA technology. It has higher risk of causing allergy compared to the pig insulin. It is exactly the same as insulin produced in the human pancreas. It is produced by inserting the human insulin gene into the bacteria, requires human cell for production. Human insulin produced through the rDNA technology. Human insulin produced through rDNA technology. It has higher risk of causing allergy compared to the pig insulin. No. Lower risk. It is exactly the same as insulin produced in the human pancreas. No, it is not exactly the same. It is produced by inserting the human insulin gene into the bacteria. Yes, bacteria what? Bacteria? Name of the bacteria? What? Mention it in the comment section. We have already read about it. Next. Which of these is an accurate description of the gene therapy? Accurate description of the gene therapy. A form of the site-directed mutagenesis, introduction of the cloned gene living into the living cell to cure diseases. Cloning of the disease gene in order to understand the gene disease, all of this. Now they are saying which of this is an accurate description of gene therapy. Accurate description of gene therapy. A form of the site-directed mutagenesis. It is introduction of the cloned gene to the living cell to cure disease. Yes. This is what? This is an accurate description. Why we are using gene therapy? To cure any disease in our, to cure the disease. So, introduction of the cloned gene to living cell to cure diseases. To cure the disease. Next. Identify the correct statement. ADA deficiency is caused due to the deletion of the gene for adenosine deaminase. Bone marrow transplant and enzyme replacement therapy are completely curative. Lymphocyte transformed with CADA gene are permanent cure for the condition. Identify the correct statement. We have to identify the what? Correct statement. ADA deficiency is caused, why? Due to the deletion of the gene for adenosine deaminase. Yes, this is true. Addition deficiency due to the deletion of the adenosine deaminase gene. Bone marrow transplant and enzyme replacement therapy are completely curative. No, they are not completely curative. Lymphocyte transformed with CADA gene are permanent cure for the condition. Lymphocyte transformed with CADA gene are permanent cure for condition. No. Only option 1 is correct. Correct. Next is the first clinical gene therapy trial was given in 1990 to a 4-year girl with alpha-1 antitrypsin deficiency, cyst fibrosis, adenosine deaminase deficiency, chronic myeloid leukemia, adenosine deaminase deficiency. This will be the correct option. The first clinical gene therapy trial was given in 1990 to a 4-year-old girl. It was what? She was suffering from adenosine deaminase deficiency. Next, gene therapy for ADA deficiency have been tried with the help of which of the following vector? Gene therapy for ADA deficiency. For ADA deficiency, we used microinjection, liposome, a bacteria and a retrovirus. Which of the following? For ADA deficiency, we used retrovirus. For ADA deficiency, we used what? Retrovirus. We used retrovirus. Next, the possible permanent cure for SCID can be enzyme replacement therapy, in which functional ADA is given to the patient by injection, bone marrow transplantation, a functional ADA-cDNA using a retroviral vector introduced into lymphocyte, gene isolated from marrow cells producing ADA is introduced into the cell at early embryonic stage. Yes. For SCID, the possible permanent cure, what we can do is gene isolated from the marrow cell producing ADA is introduced into the cell at the early embryonic stages. Next, ADA is an enzyme which is deficient in a genetic disorder, SCID, which is what is the full form of ADA? Full form of ADA is adenosine deaminase. Next, consider the following two statements. Retroviruses are useful as vector in gene therapy. They can produce useful gene product in significantly greater quantity than a human cell can. Retroviruses are useful as vector in gene therapy. Yes, this is a true statement. This is a true statement. They can produce useful gene product in significantly greater quantity than a single human cell can. No, not more than single human. This is a false statement. So only option 1 is correct. Next, the site for the production of the ADA in the body is what? ADA is lymphocyte. The site of the production of what? The site of the production of ADA. The site of the production of ADA in our body. In our body is what? Lymphocyte. Next, assertion reason. Assertion. It says delivery of the ADA cDNA into the lymphocyte is not a permanent cure for the ADA deficiency. Reason, because lymphocyte do not express ADA gene. Lymphocyte express ADA gene. Lymphocyte express ADA gene. This is wrong. So statement reason is false. Delivery of the ADA cDNA into the lymphocyte is not a permanent cure for the ADA deficiency. Yes, this is true. Delivery of the lymphocyte is not a permanent cure for ADA. When it is a permanent cure, it is a permanent cure if introduced into the embryonic stage. When introduced into the embryonic stage. Okay, next. So assertion is true, but reason is false. Next, statement 1 and statement 2. Statement 1 says, in some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement theory. Both these approaches completely curative. No, both these approaches, in first approach, it is cured by bone marrow transplantation. Yes. Statement 1 is correct. In some children, ADA deficiency can be cured by bone marrow transplantation, and in others, it can be treated by the enzyme replacement therapy. Yes, both these approaches are completely curative. Both these approaches are not completely curative, but the first statement is true. If we read it independently, we will see in some children what happens? ADA deficiency, it can be cured by what? It can be cured by bone marrow transplantation, and in other people we can use the what? Enzyme, ELISA, okay, enzyme replacement therapy. And in statement 2, it says that both these approaches are completely curative. No, both these statements are not completely curative. So option statement 1 is correct, but statement 2 is incorrect. Statement 1 is correct, but statement 2 is incorrect. Next, consider the given two statement. First assertion, enzyme replacement therapy in ADA deficiency is not completely curative. Yes, this is a true statement. Reason, because enzyme replacement therapy can induce several toxic reaction in the host. It can what? It can induce several toxic reaction, because enzyme, it can make many severe toxic reaction into the host. This is a what? False statement, and this is a true statement. It is not completely curative. It can induce several toxic. Okay, so let's continue with question number 55. Question number 55, it says, Consider the given two statement. Assertion is given and reason is given. Assertion says enzyme replacement therapy in ADA deficiency is not completely curative. Enzyme replacement therapy can induce several toxic reactions in the host. Now, what does the question says? The question says that enzyme replacement therapy in ADA deficiency, it is not completely curative. This is a true statement because why? Because the enzyme replacement therapy, it is actually not a completely completely curative, you know, it is not actually completely curative. So the enzyme replacement. Okay, so let's continue with question number 55. It says consider the given two statements. Assertion and reason is given. Assertion says enzyme replacement therapy in ADA deficiency is not completely curative. Enzyme replacement therapy can induce several toxic reactions in the host. Now, the assertion it says that enzyme replacement therapy, okay, like ELISA, like what? Like the ELISA, PCR, and all, okay. So, enzyme replacement therapy in the ADA deficiency, it is not completely curative. This is a true statement. It is actually not completely curative, okay. And they are saying that the reason is it's not completely curative because the enzyme replacement theory can induce several toxic reactions in the if. Now use your common sense. If it would induce several toxic reactions in the host, would we prefer for this kind of therapies? No, we wouldn't prefer any kind of therapies like this. It is not it do not induce any kind of toxic reaction, but yes, it is not completely curative. We agree with this statement, but it do not induce any kind of severe toxic reaction in the host body. So this is a false statement, okay. So we can say that assertion is true but reason is false. Next, ADA's primary function in the human is what? ADA's primary function in the human is to develop and maintain the immune system. अभी रात के 1 बज रहे हैं, 1 बजने वाला है, तो मेरा वॉइस में थोड़ा सा मतलब ऐसा हो रहा है कि थोड़ा सा लो हो रहा है. प्लीज़ मैनेज विथ माई वॉइस बिकॉज घर पे सब सो रहे हैं, तो आई होप यू कैन अंडरस्टैंड. Okay, let's continue. ADA primary function in the human is to develop and maintain the immune system. So what is ADA primary function? Why is ADA so necessary in our body? Because it develop and maintain the immune system of the human beings. Immune system develop and maintenance by the ADA. Okay. Now, next. Adenosine deaminase deficiency is a disorder caused due to what? Adenosine deaminase deficiency is a disorder. It is caused due to deletion of the gene for adenosine deaminase. Okay. Now, why does this deficiency occur? Adenosine deaminase deficiency. It is a disorder. It is a disorder. It is caused due to what? Due to the deletion. The body deletes this gene, okay. This is a good gene, but the body misinterprets it and deletes this kind of good gene, okay. So deletion of this gene for adenosine deaminase, okay. Next, the biggest advantage for using recombinant DNA technology, PCR, ELISA, over conventional diagnostic technique like serum and urine analysis will be what? Early detection. We can do early detection by all this kind of techniques. Next, assertion reason again. PCR has revolutionized the modern diagnosis and allows early diagnosis in many cases because PCR can detect very low amount of DNA in a sample. I think the assertion is also true, the reason is also true, and reason is the correct explanation of assertion. Yeah, both are correct, and this is done. ELISA is based on the principle of what? Antigen antibody interaction. With the help of ELISA, infection by pathogen can be detected by the presence in a sample of antigen of the pathogen and antibody synthesized against the pathogen, both 1 and 2. Antigen antibody, right? A probe, which is a molecule used to locate homologous sequence in a mixture of DNA or RNA molecule, could be either DNA or RNA. So a probe can be either DNA. Probe can be either DNA or RNA. Okay, probe can be either DNA or RNA. Okay, next, transgenic animal. Transgenic animal are the animals that have had their DNA manipulated to possess and express extra gene or the foreign gene. For the foreign gene, the DNA is manipulated. Okay, the DNA transgenic animals are the animals that had their DNA, that had their own DNA, but we manipulated. Okay, we transformed the DNA to express a foreign gene. To express a what? Foreign gene. Over 95% of all the transgenic animal is mice. We already know this. Okay, next, transgenic animal used to test the safety of polio vaccine are mice. Next, transgenic cow Rosie produces milk that is protein enriched. Okay, Rosie, it was protein enriched. Alpha 1 antitrypsin was produced in a transgenic sheep Tracy. This enzyme is used in the treatment of emphysema. In the treatment of what? Emphysema. At present, transgenic animal cannot be used for xenotransplantation. We can use it for harvesting, biological product testing, vaccine safety, and study of human disease, but we cannot use it for what? Zero what? Transplantation. We cannot use this. Okay. Next, transgenic mice are used to test the safety of the polio vaccine. They could replace the use of what? They could replace the use of monkeys. They could replace the use of monkeys. Okay, monkeys are also used for tests and all, but mice, they are more in number, more in population, so we prefer mice nowadays. Okay, and they show 100% result. Okay, next, assertion reason again. Transgenic animal can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. For example, study of the complex factor involved in growth, such as insulin-like growth factor. This is a true statement because by introducing genes from other species that alter the formation of this factor and studying the biological effect that result, information is obtained about the biological role of the factor of the body. Say, both assertion and reason are true, and reason is the correct explanation of assertion. Now, what does it say? Let's read it again. The assertion says that transgenic animal, they can be specifically designed to allow the study of how genes are regulated. This is true. Genes are regulated. We design the gene in such a way to allow the study of other foreign gene, isn't it? And how they affect the normal function of the body, and the development. For example, study of the complex factor involved in the growth, such as insulin-like growth factor. Next is, by introducing gene from other species that alter the formation of this factor and studying the biological effects that result, information is obtained about the biological role of the factor of the body. It says, introducing these foreign genes from other species that alter the formation of this factor and studying the biological effect, मतलब उसका biological effect मतलब study करने के लिए, फिर उस जीन में कैसा अल्टर हो रहा है, मतलब कैसा चेंज हो रहा है, कौन सा फैक्टर इफेक्ट कर रहा है, तो ये सब स्टडी करने के लिए हम क्या करते हैं, बायोलॉजिकल रोल क्या है, मतलब क्या फंक्शन हो रहा है बॉडी के अंदर हमारे. हम ह्यूमन लेके तो मतलब टेस्ट वगैरह नहीं कर सकते, तो हम माइस के ऊपर या फिर कोई भी ट्रांसजेनिक एनिमल के ऊपर क्या करते हैं, टेस्ट करते हैं और देखते हैं कि कौन सा कैसा हमारे बॉडी के अंदर रिएक्शन होता है, ठीक है। ओके, नेक्स्ट, इफ सक्सेसफुल एंड फाउंड to be reliable, what GM animal would replace the use of monkey to the safety of batches of the oral polio vaccine? Of course, the mice. Consider the given two statement, assertion, reason again. The milk produced by the first transgenic cow was rosy, was nutritionally more balanced product for human babies than the natural cow milk, because the first transgenic cow rosy produced human lactose enriched milk. No, it did not produce human lactose enriched milk. This is a false statement, but this is a true statement. This is a true statement. Okay. The first transgenic cow, Rosie, produced human lactose enriched milk? No, it was protein enriched. It was protein enriched. We have already read it here. See, let me show you. See, the transgenic cow Rosie produced milk that was protein enriched, okay? Protein enriched. And transgenic sheep was Tracy. Alpha 1 antitrypsin by the transgenic sheep Tracy, alpha 1, what? Alpha 1 antitrypsin used in the treatment of what? Used in the treatment of emphysema. And this protein rich milk, where is it used? It is used for nutritionally more balanced product for human babies, okay. So our assertion is true and reason is false. Next, an animal that has had foreign genes intentionally introduced into its genome using the genetic engineering technique will be called as what? An animal that has had a foreign gene intentionally introduced into its genome using the genetic engineering technique will be called as transgenic. Why? Because the animal has had foreign gene from another species we have induced into its body intentionally, okay, intentionally to test and for different kind of test, then genetic engineering technique. All this kind of animal are known as what? Transgenic. Recombinant, no, not recombinant, it is transgenic, okay, remember. Okay, next, which statement about the transgenic animal used in testing vaccine safety is correct? Transgenic animals are primarily used because they reproduce more quickly. They are genetically modified to have immune system that mimic human responses. This is the true statement used for testing. Why do we use transgenic animal? Why? Because they can easily mimic, this word is very important, they can mimic human responses, okay, they can genetically mimic human responses. Next, transgenic animal less expensive to maintain compared to transgenic animal. They are used because they do not require any ethical, no, they require ethical approval for experiment and all. Our main answer is what? They are genetically mimicking what? They are genetically copying the human response, okay, clear? Okay, next, which of the following is not applicable on genetically modified organism currently in use? Transplantation of the heart, kidney and liver, this is not possible. Next is what? Ethical issues. The name Genetic Engineering Approval Committee, GEAC, has been changed to Genetic Engineering Appraisal Committee. Next, bioprospecting, bioprivacy, bioremediation and biofortification. Now, see, first, bioprospecting, okay, bioprospecting, it will be what? Bioprospecting means the process of the discovery and commercialization of the new product based on the biological resources. The process of what? The process of discovery and commercialization of new product based on the biological process of discovery. New discovery. Prospect means what? Prospect means discovery. New discovery. Okay, So the process of discovery and commercialization of the new products based on the biological resources. Next is biopiracy. The word piracy says what? The word piracy comes, see, we have read about English word pirates. Who are pirates? पायरेट्स वो होते हैं जो समुद्र में चोरी करते हैं, उसको हम पायरेट्स बोलते हैं, isn't it? और पायरेसी मतलब क्या? चोरी करना. क्या? चोरी करना, isn't it? तो पायरेसी मतलब what? Biopiracy means the exploitive patenting of already widely used natural resources such as plant varieties by commercial entities. The exploitive patenting of already widely used natural resources, okay, such as plant variety by commercial entities. मतलब दूसरे देश में हमारे सपोज हल्दी एंड ऑल, हल्दी की एंटीसेप्टिक जो गुण होते हैं वो बाहर के जो कंट्रीज़ हैं वो लोग लेके जाते हैं और बोलते हैं वो लोग मतलब उन लोगों का क्या हुआ है, पेटेंट है, तो उन लोगों ने हमसे चोरी किया है, क्योंकि इंडिया में पहले आ चुका था ये सब चीज़ें, ठीक है, लेकिन वो उन लोगों ने चोरी किया और बोल दिया कि उन लोगों ने खुद निकाला, तो वो सब गलत है. नेक्स्ट, बायोरेमिडिएशन. What is bioremediation now? Bioremediation is a process that uses microorganisms or their enzymes to treat polluted sites for regaining their original condition. It is a process. Bioremediation. Remedy मतलब क्या होता है? Remedy मतलब क्या बोलते हैं? Remedy मतलब क्या बोलूँ मैं अभी? The word रेमेडी का ये बोल सकते हैं हम क्योरनेस बोल सकते हैं ना, तो ऐसा बोल सकते हैं कि क्योरनेस ऑफ पेन, हमारा कोई दर्द हो रहा है तो वो सब क्योर करना. तो बायोरेमिडिएशन मतलब क्या होता है? मतलब बायोलॉजी रिलेटेड, मतलब हमारे बॉडी में जो एंजाइम्स है, ठीक है, द प्रोसेस दैट यूजेस microorganism or their enzyme to treat polluted site for regaining their original condition. ये सब करने के लिए हमें क्या करना पड़ता है? बायोरेमिडिएशन करना पड़ता है. इट इज़ अ प्रोसेस दैट यूजेस माइक्रोऑर्गेनिज्म और देयर एंजाइम्स. ओके, दैट प्रोसेस दैट यूजेस माइक्रोऑर्गेनिज्म और देयर एंजाइम टू ट्रीट polluted sites for regaining their original condition. चाहे वो हमारा बॉडी हो जाए, चाहे वो किसी भी जगह की बात कर ले, ठीक है. तो फॉर रिगेनिंग देयर ओरिजिनल कंडीशन में लाने के लिए हमें क्या करना पड़ता है? बायोरेमिडिएशन करना पड़ता है. नेक्स्ट इज बायोफोर्टिफिकेशन. तो बायोफोर्टिफिकेशन क्या बोलता है? द प्रोसेस बाय विच न्यूट्रिशनल क्वालिटी ऑफ द फूड ग्रुप इज इम्प्रूव्ड थ्रू एग्रोनॉमिक प्रैक्टिसेस, कन्वेंशनल प्लांट ब्रीडिंग और मॉडर्न बायोटेक्नोलॉजी. सो बायोफोर्टिफिकेशन क्या होता है? It is a process by which the nutritional quality, biofortification मतलब nutritional quality of the food group. Fortification मतलब food related. Okay, fortification means what? Food nutritional value. Okay, the process by which food of the group is improved or through agronomic practices, conventional plant breeding or modern biotech. So ऑप्शन विल बी ए बी सी डी. डायरेक्ट सीख लो तो अच्छा है, ऑप्शन पे न मत जाओ, यहाँ पे डेफिनेशन सीख लो, हो जाएगा काम. नेक्स्ट, आइडेंटिफाई द इनकरेक्ट स्टेटमेंट. नाउ वी हैव टू आइडेंटिफाई द इनकरेक्ट स्टेटमेंट. लेट्स रीड. देयर आर 27 डॉक्यूमेंटेड वेराइटी ऑफ बासमती ग्रोन इन इंडिया. बायोपायरेसी का एग्जाम्पल हो गया, ठीक है. तो देयर आर 27 डॉक्यूमेंटेड variety of basmati grown in India. This is a true statement. 27, remember this number 27, and remember basmati, ठीक है. Okay, next, in 1997 an American company got the patent rights on the basmati rice by Biopharmacy. See, American company लेके चला गया, ठीक है, got the patent rights of the basmati rice by Biopharmacy. Next, Indian basmati was crossed with semi-dwarf variety and claimed as an invention or novelty. There are an estimated 200 variety of rice in India alone. No. There are an estimated how many variety? कोई तुक्का लगा सकता है, मतलब कोई बोल सकता है कि कितने वेराइटीज़ हैं यहाँ पे? ऑप्शन दे दो. चलो ऑप्शन दे देती हूँ. ऑप्शन दे दो. चलो ऑप्शन दे देती हूँ. पहला ऑप्शन है 200, एक तो ये ऑप्शन हो गया. दूसरे 3 ऑप्शन देती हूँ, ठीक है. एक है 2000. नंबर सी ऑप्शन सपोज 20,000. कौन सा वाला होगा? 200, 20,000 या फिर दिस सेमी. बताओ कौन सा वाला होगा? देखो ये वाला होगा आंसर, ठीक है. ऑप्शन सी विल बी द ऑप्शन. ओके, so there are estimated how many variety? 200 variety of 200, no, 20,000 200,000, sorry, गलत हो गया गाइस. 200,000 okay, 200,000 varieties. Okay, so there are estimated of how much? 200,000, not 200 varieties of rice. 200,000. Next, exploring molecular, genetic and species level diversity for the products of economic importance unethically appropriated and without fair compensation to indigenous people is known as biopiracy. You already know, without fair compensation, chori ka maal. Okay, so it will be what? Biopiracy. The last, biopiracy is, what is biopiracy? We have already learned the definition. It is exploitation of a region's biological resources or indigenous knowledge unethically and without providing fair compensation. Consider the given two statements, statement 1 and statement 2. The biological significance of manipulation of living organism by human is important. Genetic modification of organism can have unpredictable result when such organisms are introduced into ecosystem. Now what does statement 1 says? Statement 1 बोल रहा है कि biological significances of the manipulation of the living organism by human is important. Yes, this is very important because biological signification of significance of manipulation by living organism by manipulation of the living organism by what? Human is important. You have to give the biological significance. You cannot manipulate any innocent animal for simply, isn't it? So you have to give the biological significance for the manipulation of the DNA. Okay, manipulation of the DNA, that is transgenic animal. Why are you making transgenic animal? You have to give some reason. So humans should have such kind of biological reasons that they are manipulating the genes of the living organism. Statement 2, what does it say? This is true statement. Okay, second statement, genetic modification of the organism can have unpredictable result when such organisms are introduced into the ecosystem. Now what they are saying, that genetic modification of the organism, they can have unpredictable result. Always predictable? No. Then why are we doing tests, right? So genetic modification of the organism, they can have unpredictable result when such organisms are introduced into the ecosystem. So statement 1 and statement 2 both are correct. Last question, how many documented varieties of basmati are grown in India? we have already read about it. It will be, answer will be how many? 27. Okay, so that's it for today. Okay, as I had said that I would give the answer of some question that you will mention in the comment section. Let me say the answers now. Let's see if we had remembered. First is this: human insulin produced through recombinant DNA technology. Insulin in the bacteria, what? Bacteria will be E. coli. Bacteria is E. coli. Bacteria E. coli. Okay, and the next one is, yes, this. Okay, Agrobacterium tumefaciens. Where was it used? As an injection in BT, isn't it? In BT, to induce BT, to induce BT in tobacco, in tobacco. Okay, so to induce BT in tobacco, Agrobacterium tumefaciens were used. So that's it for today, and it's my first video, so please like, शेयर एंड सब्सक्राइब अगर वीडियो पसंद आए तो, नहीं तो कोई प्रेशर नहीं है. ओके, आई विल इम्प्रूव मोर. प्लीज़ कमेंट कर देना कि मुझे क्या इम्प्रूवमेंट करना चाहिए, और आई विल श्योरली ट्राई. दिस इज़ माई फर्स्ट वीडियो. प्लीज़ सपोर्ट गाइस. दिल से चाहती हूँ कि मैं जो पढ़ाई कर रही हूँ वो आप लोगों के साथ भी शेयर करूँ, साथ में ग्रो करें. That's it. That's all for today. Thank you.